Welcome to this explainer. Today, we're putting on our genetic detective hats to uncover how tiny molecular typos in our DNA completely shape human traits and human health. I mean, have you ever actually stopped to wonder why siblings can look so incredibly similar or sometimes just wildly different? Or how a single microscopic glitch in our biological code can literally echo across generations? Well, we're going to decode those tiny typos that write the story of who we are. Here's our quick roadmap for today. We are going to start with the nature of mutations, move right into pedigree analysis, decode some sex-linked disorders, and then wrap the whole thing up with a deep dive into the genetics of sickle cell. All right, section 1, the nature of mutations. When the genetic code changes, let's look at what actually happens when our biological instruction manual gets altered. So think of your DNA as this massive 3 billion letter book. And most of the time, it's copied flawlessly from one generation to the next. But occasionally, a little typo slips in. We call these typos mutations. And the crazy thing is, they change not just our hidden genetic code, our genotype, but they can also alter the physical traits we actually see out in the world, our phenotype. Now, to really grasp the scale of these errors, imagine the difference between, say, a single wrong letter, which is a point mutation, the culprit behind conditions like sickle cell anemia, and having entire chapters of that book just ripped out or glued in backwards. Those are chromosomal aberrations. We're talking massive, structural, chaotic events involving the absence, excess, or just totally abnormal arrangement of entire chromosomes. You'll actually see this frequently in cancer cells. And it's not always just a random copying error either. External forces, which we call mutagens, can directly induce these genetic mutations. We're talking chemical and physical factors. Even something as common as UV radiation from a sunny day at the beach can physically strike and alter your DNA. Pretty wild, right? So this brings us to a pretty fascinating mystery. Since we obviously can't perform controlled genetic crosses on humans to see how traits are passed down, like we aren't Gregor Mendel working with pea plants here, how on earth do we actually track these genetic typos across generations? Which leads us to section 2, pedigree analysis. The Genetic Detective's Toolkit. Okay, so because those controlled human crosses are completely off the table, our analysis has to rely entirely on studying family histories. By drawing out family trees over several generations, using specific symbols like squares for males, circles for females, and shading them in to track a particular trait, we essentially create this powerful visual map. This pedigree analysis is absolutely crucial. It provides us with a really strong tool to accurately trace abnormalities, diseases, and specific traits as they move through a family line. All right, moving on to section 3, decoding sex-linked disorders, traits tied specifically to the X chromosome. Take conditions like hemophilia, which is a disorder where blood doesn't clot properly, or red-green colorblindness. Because these specific recessive typos live on the X chromosome, our pedigrees reveal a very clear pattern of inheritance. They usually jump from a carrier mother, who actually doesn't exhibit the disease herself, directly to her male progeny. And this dynamic perfectly explains why men are just overwhelmingly affected here. I mean, look at these stats. Colorblindness occurs in about 8% of males, but a tiny 0.4% of females. The biological reason is actually brilliantly simple. Males possess only one X chromosome. They literally have no biological backup. Females, on the other hand, possess 2 X chromosomes, meaning they'd need the mutation on both of them to actually be colorblind. Otherwise, they just remain unaffected carriers. Finally, section 4, the genetics of sickle cell, a molecular deep dive. Now we're zooming into Mendelian disorders that are autosomal linked, so mutations on the non-sex chromosomes. This group includes conditions like phenylketonuria and thalassemia. But for our focus today, let's look at sickle cell anemia. The math on this is really striking. Even if 2 parents appear perfectly healthy, if they both carry just one hidden copy of the mutant gene, making them heterozygous carriers, there is a flat 50% coin flip during transmission that they'll pass that mutant gene down to their progeny, inherit the mutant gene from both parents, and the child becomes homozygous and they'll actually exhibit the disease. It is honestly incredible how tiny this glitch is at the molecular level. Step 1, it all starts with a single base substitution at the 6th codon of the beta-globin gene. Just one letter swapped out, JG becomes GUG. Step 2, that single typo swaps out one amino acid building block, replacing glutamic acid with valine at the 6th position of the beta-globin chain. Because of this tiny change, step 3 happens. the mutant hemoglobin undergoes polymerization, basically clumping together into rigid chains whenever oxygen is low. And finally, step 4. This clumping physically warps the red blood cell, changing its shape from a healthy, flexible biconcave disc into a stiff, sickle shape. Literally, one single letter change completely alters the architecture of an entire cell. So as we wrap up this explainer, we're left with a pretty profound question. If a single letter typo in our DNA can cause that much structural change, how does our growing ability to read this code empower us to better predict, track, and ultimately treat the genetic disorders of our future? By tracking pedigrees and mapping these molecular glitches, we are no longer just guessing at heredity. We finally have the blueprint. The real question is, what are we going to build with it? Thank you so much for joining me to explore this fascinating genetic landscape. Keep your curiosity alive, and as always, keep learning.